warm welcome to Bioinorganic Chemistry course. Let me introduce first myself. This is Dr. S. P. Rath, professor in the Department of Chemistry at IIT Kanpur. And the Bioinorganic Chemistry is a meeting point of chemistry and biology, and this has been designed to understand the role of inorganic elements in biological processes. And this bioinorganic chemistry is a highly interdisciplinary subject and meeting point of various fields like inorganic chemistry, physics, biochemistry, microbiology, nanotechnology, physiology, pharmacology, etc. The main avenues of this study would be understanding the role of naturally occurring inorganic elements in biology. Metal ions at the core of biomolecules control many key processes. Understanding structure and function of the metalloenzymes and the roles of metal ion therein. Practical application of the knowledge gained and use of inorganic molecules as probes and drugs. For example, cisplatin, aronophine, cardiolite, several such examples are there. I will now take a joyride to highlight what would be showcased in the next few hours of my journey in this short course of bioinorganic chemistry. Please note that only few selected topics will be chosen to cover during this short time. The most abundant element in the earth crust is oxygen, and you see there can be many chemical reactions with oxygens. However, all these reactions are thermodynamically favorable and exothermic. However, it never happens just like mixing. The redox chemistry of molecular oxygen is shown over here, and the most important reactions is when dioxygen converts to water with the help of 4 proton and 4 electron with a redox potential of 0.815 volt. The reduction potential of the 4 electron reduction of dioxygen is a measure of great oxidizing power of dioxygen. If this being so, then what protects vulnerable species from the full oxidizing power of dioxygen? The percent of atoms in human body is shown over here. As you can see that hydrogen is around 62.8%, oxygen 25.4, carbon 9.4, nitrogen 1.4, and other elements together is just 1%. At first sight, the idea of inorganic chemistry associated with life may appear to be rather narrow field of study, as we tend to think of living matter as being just organic. However, it is a fact that without certain inorganic elements, no organism could exist. The term bioinorganic, which is a composite of biology and inorganic, is used to describe the occurrence and properties of inorganic elements in living system, and these elements are extremely important for our day-to-day life. I have shown here the periodic tables of element, and as one can see that some of these elements are highlighted blue, green, pink, brown. The brown color is for the elements which are present in bulk amount in biological system. Then the blue one, which is believed to be the essential for a wide range of bacteria, plants and animals including humans. Then green, which is for elements that may possibly be essential for some species, whereas pink is for those elements which are believed to be essential for some species. As you can see that most of the elements are not even used in biology. Now, we will discuss that how nature has selected only certain elements from large number of elements present in the periodic table. Now, what role does the metal ions play? The metal ions are present at the active site of biomolecules, especially metalloenzymes, and they have been doing as some functional role as also structural role. However, metalloenzymes without metal ions are simply inactive. I will now show you an example of a metal ion in a functional role. Say carbonic anhydrase is actually responsible for conversion of carbon dioxide and water to bicarbonate NH plus, and this is the active site structure of carbonic anhydrase, where you can see that 3 histidine are ligated and the 4th position is occupied by weak water ligand. The uncatalyzed reaction is too slow, is just 7 into 10 to the power minus 4 second inverse. The above reactions, when it is catalyzed by carbonic anhydrase, is 10 to the power 7 times faster. Turnover number of this reaction is 10 to the power 6 second inverse, huge turnover number. We will discuss that how this enzyme actually increases the rate of these reactions. X-ray structure of carbonic anhydrase is shown over here. You see that there is a huge protein chains wrapping around this molecule, and inside this protein this zinc ion is sitting, and once we remove this protein chain, you can see the active site structure of this carbonic anhydrase. Now, you will see that how the metal ion is responsible for its conversion to bicarbonate. This is the protein structure with carbon dioxide as a substrate. You see that carbon dioxide is sitting over there. Again, once I remove this protein chains, you can see that carbon dioxide is sitting close to metal center. And this is also the protein structure of carbonic anhydrase, and formation of bicarbonate is clearly visible. And once I remove this protein chains, you can see that the bicarbonate is formed over here, and this is ligated to the zinc metal center. So, you can see yourself that how zinc is responsible for this transformation of carbon dioxide to bicarbonate. I will now show you a metal which is in a structural role, zinc finger protein. Now, zinc finger proteins are essential to recognize and bind to DNA. They have finger like projection which fit inside the grooves of the helix. As one can see that this is the polypeptide chain, and zinc is sitting inside with ligated with nitrogen and sulfur. Now, once you remove the zinc, the entire structure collapses. So, this suggests that how crucial the zinc metal ion is. Now, question is that how the elements transport it across the cell membrane. So, you can see that the elements in high concentration comes through the biological membrane to the low concentration very easily across the concentration gradient. This is passive transport. Now, if the elements have to go to the opposite direction, like from low concentration to high concentration, you must need an energy to do that, because it is not spontaneous, though this is called active transport. Now, elements like iron, zinc, copper, molybdenum comes inside the cell against the concentration gradient, since their concentration increases by many thousand folds inside the cell. It would be interesting to discuss how the specific receptor proteins allows only some selected elements to come inside the cell. So, it can be passive diffusion, it can be through an ion specific channel, but it would be very selective, so that not every element can come inside the cell. So, we will be talking about how ions are selected by the specific receptor proteins. For example, this potassium channels is shown over here, and this is very much specific for the potassium, and it only allows potassium to go through this channel. And Roderick Mackinnon was awarded the Nobel Prize in Chemistry in 2003 for structural and mechanistic study of ion channels. So, we will be discussing all this in details in my subsequent lectures. Now, I have shown here the symptoms of element deficiency in humans. As you can see that large number of deficiencies or diseases can happen once you have a deficiency of a particular element. For example, calcium, that is retarded skeletal growth, magnesium, iron, zinc, copper, molybdenum, cobalt, nickel, chromium, silicon, fluorine, iodine, selenium, arsenic, all these elements have to be present within a certain concentration range. If it is present in more concentration or less concentration than the desired amount, then one would be having diseases, and some of these symptoms are tabulated over here. Now, some of these effects of metal deficiencies are shown over here, and if you have the deficiency, you have to take medicines to supplement those metals and go to the doctor, and doctor will write these medicines indeed. Now, two most important biological reactions are shown here. For example, that we take dioxygen, and dioxygen is being circulated in the blood, and it releases lots of energy and carbon dioxide during the respiration. And this carbon dioxide transported through blood goes out of our body, and it is being utilized by green plant, and in presence of light they undergo a photosynthesis reactions, and it liberates oxygen which we inhale. So, this is being the cycle of two important reaction, one is photosynthesis, another one is respiration. Now, the dioxygen carrying proteins are shown here in hemoglobin and myoglobin in our body, and as you can see that deoxyhemoglobin and myoglobin, which is 5 coordinated, convert to 6 coordinated upon dioxin binding, and iron 2 getting oxidized to iron 3, whereas dioxin getting reduced to superoxides. Now, hemoglobin and myoglobin are responsible for this dioxin transport and storage in our body. However, hemoglobin is tetrameric in nature, as you can see that these heme centers are here 1, 2, 3, 4. However, huge proteins are wrapping around these heme centers. Similarly, myoglobin, there is only one heme centers, and basically the structure of this heme center is shown over here. This being the case, now we need to understand that how nature designed these molecules. The hemoglobin selected for transporting oxygen, while myoglobin stores oxygen. Myoglobin is monomer, while hemoglobin is tetramer of the same structural units, discriminating oxygen from other available gases in the environment. So, we will be talking all this and try to understand the nature's design. For example, this distal histidine, which is not ligated directly to the iron center, is responsible for discriminating between carbon monoxide and oxygen. As one can see that this distal histidine, while facilitating the angular coordination of dioxygen comfortably, while the perpendicular and linear carbon monoxide moiety cannot bind like this, it is forced to have in a angular fashion. In the respiration, the most important transformations that happens is dioxin convert to water with the help of 4 proton and 4 electron. Dioxin completely reduced in this way. However, if it is reduced partially, what would happen? So, dioxin after 1 electron reduction converts to superoxides, superoxide then convert to peroxides, peroxides after reduction converted to water and OH radical, and this OH radical converts to water. As you can see that partial reduction produces superoxide, peroxides, hydroxy radical. These are actually called reactive oxygen species and very harmful for our biological system. And this reactive oxygen is responsible for aging and age related disease in our body. So, I have shown here how reactive oxygen species actually changes our faces, and these antioxidants are also there to fight against this reactive oxygen species, and some of these food items are shown here which are strongly antioxidant which destroy those ROS, that reactive oxygen species. And this is what one can use at home without losing much. You can stay thin also at very old age. Now, secret of anti-aging agent is superoxide dismutase. As one can see, the superoxide getting converted to oxygen and O2 2 minus peroxide. However, this is a dismutation reaction, means this is a spontaneous process, thermodynamically feasible, delta G negative. Now, this being the case, why nature designed enzyme called superoxide dismutase? This is because the rate of these transformations has increased enormously. It becomes 2 into 10 to the power 9 mole inverse second inverse, and that is the reason why nature has designed an enzyme, because superoxides are extremely poisonous. However, the superoxides also produce peroxides, which is another poisons. Now, this peroxides is undesired and also extremely harmful. However, nature utilized this undesired product, harmful product, to something very useful, and some of the enzymes such as catalase, peroxidase, chloroperoxidase, lignin peroxidase, cytochrome superoxidase, there are lots of enzymes which actually converts this undesired intermediate into something very useful molecules in our biological system. Now, what could be the better waste management than this? Cytochrome P450 is another very important enzymes in our body, which is responsible for oxidative metabolism of drugs, steroids and carcinogen. It actually protects our body from toxification and hydrolyze undesired compound, so that it can excrete through our body. Now, this cytochrome P450, catalase and peroxidase all go through a highly reactive intermediates, that is called iron 4 oxo species, and they are all similar, only difference is their axial coordination. The sixth position is occupied, in some cases it is histidine, in some cases it is tyrosine, in some cases it is cysteine. However, their structures are indeed very similar. Now, it is very surprising to see that their activity is very different. For example, in cytochrome P450, it is actually responsible for oxo transfer to substrate, while catalase and peroxidase, it is electron transfer to substrate. Now, it is interesting to see that how design principle makes all these differences in reactivity. We have also learned from nature and implemented the knowledge gained for the betterment of our day-to-day life. Catalase and peroxidase at resting state is like this, and one of our friend from US has made this bio-friendly catalyst thermal, which is doing miracles in cleaning large number of pollutants from our environment. He also indeed earns huge amount of money out of that. The active form of this catalyst is the same for both biology and biocatalyst. This is an alternative based on nature's oxidant, oxygen or hydrogen peroxide. Now, as I have discussed that in our body, hemoglobin and myoglobin is responsible for oxygen transport in our body. Now, what is exactly happening, that iron 2 converted to iron 3, while dioxygen reduced to superoxide, and once this dioxin leaves, this iron 3 converts back to iron 2 again. So, this is completely reversible, which is happening all the time in our body. Now, dioxin binding slowly causes irreversible oxidation of iron 2 to iron 3 in deoxyhemoglobin, and in that way the overall efficiency of hemoglobin decreases over the time. Now, what is the nature's solution for it? Yes, nature has a solution. So, heme decomposition and regeneration of the blood is what is the solution. And we all know that red blood cell last only for 120 days. So, this is what that heme decomposition process catalyzed by heme oxygenase, like hemoglobin converted to biliverdin, bilirubin, which is yellow in color, however insoluble, then bilirubin converted to bilirubin diglucuronide, and which excrete through urine. So, this is what is happening all the time, and it is better that you donate your blood, otherwise it undergo this types of decomposition process all the time. As I have already said that red blood cell has lifespan of only 120 days, and after that it decomposes. So, this is what is happening, and it is a spontaneous process. If you have disbalance, then this bilirubin, which is yellow in color, will be deposited on the body and creates this jaundice. You can see this normal person is shown over here. However, the person which is having jaundice, which is his body color turns yellow, and you can see this is a very common disease in our life if it is not spontaneous. Now, I will be talking that why the rate of this electron transfer is so fast in some of these enzymatic proteins or enzymatic system. I am showing here a blue copper protein, an example where rate of electron transfer is increased to 10 to the power 8 to 10 to the power 10 times faster. In this blue copper protein, the rate is 10 to the power 4 to 10 to the power 6 mole inverse second inverse. However, what is happening in blue copper protein? This copper 2 converts to copper 1 and copper 1 releases 1 electron, convert to copper 2. So, copper 2 become copper 1 and copper 1 become copper 2. So, 1 takes electron, 1 release electron. So, this is what is happening. During this process, there is a huge structural reorganization energy required. Say copper 2 is 4 coordinated species, always square planar. Copper 1, which is D10 system, it prefers tetrahedral. Now, converting a square planar to tetrahedral and tetrahedral to square planar, there is a huge reorganization energy required, and this slows down the rate of electron transfer. Now, this being the case, then how the electron transfer in blue copper protein increases to 10 to the power 8 to 10 to the power 10 times faster. So, we will talk about all this in details. You must have seen that potato and apple turns brown if you cut it and keep it for few hours, and this is because that the surface is getting protected out of this brown color, and the enzyme which is responsible for this browning of the surface is tyrosinase. We will talk about in details the mechanism of this brown color and how tyrosinase actually responsible for converting this apple and potato to brown. The coenzyme B12 dependent enzymes is shown here. Basically, coenzyme B12 is a medium sized molecule with a molecular mass of about 1350 Dalton, which exhibits its characteristic specificity and high reactivity only in combination with corresponding apoenzyme. And as you can see that this vitamin B12 is the only vitamin known to contain a metal, and also now we will look at the design and nature selection of the metal and the macrocycle. This coenzyme B12 is so important that 4 Nobel Prize was awarded in 1934. Whipple, Minot, Murphy got the Nobel Prize for the discovery of anti-pernicious anemia factor, now we call vitamin B12. 1964, Dorothy Hodgkin, she solved the excess structure of this complicated molecule. 1965, Woodward for its outstanding achievements in the art of organic synthesis. 1981, Foucault and Hofmann for quantum mechanical studies of chemical reactivity. You all know that Woodward-Hofmann rules, how important it is for pericyclic reactions. Now, how inert is the nitrogen gas? This nitrogen bond dissociation energy is very high, 225 kcal per mole. Indeed, we run our reactions under nitrogen assuming that it will maintain inert atmosphere. the reaction, this nitrogen plus 3 H2 gives rise to 2 molecule of ammonia, is also thermodynamically favorable, but the kinetic barrier is too high, as one can see out of this diagram. Indeed, the industrial production of ammonia, which is popularly known as the Haber-Bosch process, is nitrogen plus 3 molecule of hydrogen converts to ammonia, which eventually transform to urea. This ammonia requires a very drastic condition of more than 200 atmospheric pressure, 450 degree centigrade temperature, and iron molybdenum catalyst. And urea is very important for our farmers. It has been used as a fertilizers, and the Haber and Bosch got Nobel Prize in 1918 and 1931 respectively. Conversion rate is only 15%. Now, in contrast, if you look at the biological catalyst nitrogenase, this is the leguminous plant, and this is the root nodules have those nitrogenase which silently fixes the aerial nitrogen, converting them to ammonia and fertilizers. So, the nitrogenase, this enzyme has this iron protein and iron molybdenum protein, and once you remove the protein, as you can see, this iron sulfur clusters and iron molybdenum clusters are actually responsible for fixing the aerial nitrogen and converting them to ammonia and other useful products. We will discuss in details in the subsequent lectures. So, the reaction which is actually happening is converting aerial nitrogen to ammonia at room temperature and normal atmospheric pressure. So, it is interesting to see that how aerial nitrogen can so easily be fixed and so easily be utilized, and we will be discussing these in details in subsequent lectures. Now, I will also talk about the inorganic molecules used in medicines, and some of these examples, popular examples, are shown over here. This is cisplatin, which is an anti-cancer drug, orofen, which is arthritic drug, cardiolite, it is an imaging agent. There are many such molecules which are used in medicine, and I will be talking all details in my lectures, and I hope our journey would be very pleasant and you will all enjoy going through this course. Thank you so much for your attention.